Magnetic nanoparticles are ideal tools for analytical sensing and nanomedicine due to their strong magnetic properties and the easy functionalization of their surface. We report here the first synthesis of a conjugate between an amino-functionalized silica magnetic nanoparticles and the Siderophore Feroxamine, along with evaluation for the capture of pathogenic bacteria. Siderophores are small organic molecules which play a key role in the iron uptake mechanism, and they are recognized by specific outer membrane receptors of bacteria.
This video displays step-by-step an efficient protocol to prepare magnetic iron nanoparticles. Then, they were coated with silica to improve their dispersion and to protect the magnetic nucleus. The resulting reactor surface was functionalized with amine groups.
Synthesis of magnetic nanoparticles conjugated with feroxamine. Add 0.5 grams of FeACAC3 in a 20 ml glass vial. Then mix with 10 ml of benzyl alcohol. Sonicate this mixture for 2 minutes. Then transfer to a heating block and heat at 180 degrees centigrade for 72 hours. Rinse the nanoparticles with 96% ethanol. And centrifuge for 30 minutes. Separate the nanoparticles